Hi, my name is Justine Whipple, and I'll be speaking to you today about the interaction between intestinal endothelial cells, or eukaryotic cells, and the prokaryotic cells of Salmonella, an invading pathogen. The date is August 14th, 2024. Let me start off by introducing the villain of this tale. Salmonella is divided into two species with a number of subspecies. They are Salmonella enterica and Salmonella bungori. These are prokaryotic cells that have a unique way of maintaining invisibility and turning our body's own defense mechanisms against us, allowing for blooms and flourishing in the moist, nutrient-rich environment of our guts. It's a gram-negative bacteria that causes abdominal pains, diarrhea, headaches, nausea, and vomiting, and can even lead to enteric fever if not treated with the right strain. This can result in deadly results for these third world countries. Humans and animals serve as reservoirs for these organisms, but the route of transmission is through contaminated water or food. Recent articles in the U.S. suggest that incidence of salmonella being carried on fresh fruits and vegetables is increasing. The rod-shaped prokaryotic cell of salmonella measures approximately 2 to 5 micrometers by 0.7 to 1.5 micrometers, with a flagella that provides for its mechanism of motility and presents different antigens, to include the protective capsule that makes bacteria resistant to the pH changes and dehydration, and presents the K or the virulence antigen. The organism also has pili on the outside of its capsule, which allows it to adhere to surfaces such as the endothelial cells within the lumen. There's virulence factors that are located within the Salmonella pathogenicity islands, otherwise known as SPIs, which is actually a cluster of genes located within the chromosomal DNA and are directly involved with the invasion process. There's a number of different virulence features that play a role in Salmonella's ability to attach, invade, and then evade the host defense mechanism. It possesses a unique ability to express different proteins at different times, which has led to them calling this Salmonella's unique ability to express different proteins at different times has been called diphasic 1 and 2 protein capability. We'll now shift over to the eukaryotic cell. The intestinal epithelial cell is a eukaryote, possessing all the normal features of a eukaryotic cell, but with specific features such as the microvilli, which are utilized in the absorption of nutrients within the lumen, as well as producing peptides that are antimicrobial within the system. There's a nice balance between its ability to maintain the commensal bacteria that helps us digest food, but also combat the pathogenic organisms such as Salmonella. These cells secrete the antimicrobial peptides and can coordinate responses against enteric pathogens, but also, like I said, have mechanisms to control signals, allowing for the continuous exposure to the commensal bacteria. Epithelial cells measure about 20 micrometers in size, so a bit larger than the Salmonella, and they include a nucleus, Golgi apparatus, cytosol, vesicles, mitochondria, and ribosomes for cell functioning. Now let's turn to the pathogenicity interaction between Salmonella bacteria, gut microbiota, and the intestinal epithelial cell. When ingested, the Salmonella pathogen is exposed to the stomach acids, which actually eradicate a number of these pathogens. The ones that remain have physiological changes to survive and pass into the small intestine, where they're exposed to the endothelial. A subset of the Salmonella can actually physiologically adapt to the environment, surviving, and then are passed on to the small intestine, where they're exposed to salts, bile, antimicrobial peptides, and the secretory immunoglobulin A. Salmonella is able to cause infection via multiple pathways that stimulate one another. It attempts to make itself invisible to the immune system and take advantage of normal responses that the body would have to combat pathogens. These pathways include the following: passive uptake by dendritic cells across this epithelial cell wall, invasion via microfold cells, or M cells, located here, bacterial replication, and secretion of cytokines resulting in inflammation or apoptosis, and a colonization of non-phagocytotic host cells that result in salmonella containing vacuoles. So here we have our non-phagocytotic host cells where this is releasing, it's entering in here, and then there's a bunch of lateral transfer of information and signaling. Inflammation occurs, and cytokines cause the inflammation and then allows this to be released back into the environment, allowing the host to flourish, or allowing the salmonella to flourish in this environment. A primary means of entry is through the microfold cells. The bacteria here induce membrane ruffles around the pathogen. So as this salmonella enters, ruffles occur, and it's then taken up. It is then pulled into around here, which allow the lymphoid, which the M cells are the lymphoid tissue, and the type 3 secretion system on the cell surface of multi-protein channels, which allows the salmonella to deliver its payload into the cytosol, allowing the salmonella to come in here, and then it's carried across into the cytosol. Salmonella can penetrate epithelial cells via protein secretion systems that allow the bacteria to be transported and modulate the cytokine expression and signals to promote bacterial replication. You'll notice, as depicted here, once a salmonella is able to cross the epithelial barrier, the bacteria can be taken up by macrophages, as notated here. Within the macrophages, they can actually initiate system disease. The salmonella is able to turn on its gene expression of genes acquired horizontally via its plasmid and modify the phagosome into a salmonella-containing vacuole and remodel the host, as you can see here, this neutrophil. So it's actually changing the host system and allowing it to not see this as a pathogen anymore. Additionally, the promotion of growth bursts of salmonella is enabled by several mechanisms as well. So first, it can compete with that microbiota for the available hydrogen. Up here, you'll notice when the salmonella is in here, there is hydrogen located within the intestinal lumen, and it's able to get that and compete. And sometimes it can outcompete that microbiota. Even if it can't outcompete that microbiota, as the other cytokines are producing inflammation, it's producing cytokines and this nitric oxide. The nitric oxide is then oxidized into nitrate, which can become part of the respiration process to fuel the growth of salmonella. Additionally, with the hydrogen sulfate that's produced from the processing of microbiota and salmonella, this is interacting with the lumen or the endothelial cells as well, producing thiosulfate. It's then oxidized into tetrathionate, which can also be used in anaerobic respiration that enables salmonella growth. So as you can see, there's multiple different pathways that's enabling these growth bursts to happen while it's modifying the actual cells here and the macrophages. Finally, it's important to note over here, as the microbiota and salmonella are produced, they're producing short-chain fatty acids, which then using these type 3 protein modulations, it actually modulates the expression, allowing the salmonella to penetrate the epithelial cells. And the other piece of this is that the salmonella are able to penetrate any leaky or any gaps within cells. So individuals that might have already immunocompromised or issues with GI may be more susceptible to the salmonella just sneaking in through different gaps. That concludes the presentation for today. If you have any questions,